Hey y'all, I'm James Few, and I did Revolutionizing Cancer Treatment, the Power of Targeted Radionuclide Therapies. So my presentation is going to focus on the lock and key mechanism behind treating breast cancer. So how can we manufacture specific drugs that act as a key in treating these cancers? So cancer remains one of the leading causes of death in the world, demanding constant innovation and treatment strategies. I'm gonna be honest, nobody likes cancer, and the topic of cancer is very hard for many people across the world generally. It's one of the hardest things to deal with, and it's one of the biggest problems that plagues the world. As you can see on the figure, the global cancer burden keeps growing. The estimated number of cancer cases and deaths worldwide per year steadily increase. It's been on a constant increase for the length of time ever since it's been discovered. The amount of cases has been increasing, and that's very problematic, and it's very detrimental to society that we keep having this burden and we keep having to deal with it time after and time out. And so it's really important that as a society as a whole that we work on finding ways of innovating and expanding upon and formulating better treatments to solve this burden. Overall, different cancers act very differently, and targeted treatments are key to improving outcomes. Here you can see the comparison between male and female and their cancer statistics, but more so specifically on the specific type of cancer deaths seen in males and females. And so, for instance, in the male population, the amount of prostate deaths is very significant in comparison to all of the other deaths experienced for the other types of cancer. But for female patients in particular, the most lethal cancer is breast cancer. And this raises a problem and it raises a question is that why are some types of cancers more treatable? Why are some more curable? And why are some more deadly in comparison to other types? And it all comes down to the idea of specificity. And so in breast cancer and in prostate cancer, it's very hard to target these very minute and very specific spots. Or even with lung cancer, we have a bunch of blood vessels, a bunch of capillaries, and a bunch of important tissues that line that area, so it makes treatment very hard. And to treat cancer overall, we need to find a way to work around these barriers and improve the treatment. And so finding ways to improve on the specificity is very important. Radiotherapy and chemotherapy are global staples in cancer treatment. Chemotherapy is essentially a systemic form of cancer treatment that involves the use of powerful drugs designed to kill rapidly dividing cells throughout the body. Since cancer cells grow and multiply extremely fast, chemotherapy is able to exploit this vulnerability and halt tumor growth and its spreading. What is listed on the right, it's pretty much administered through kind of like an IV. And so you have this chemotherapy drug and it's given intravenously throughout the body. And it flows throughout your body and it targets these cancer cells throughout the bloodstream. On the right hand side, you see a picture of someone putting a individual through a radiotherapy machine. And so how this radiotherapy works is that radiation therapy is a localized cancerized treatment, and it pretty much uses high energy radiation, such as utilizing an X-ray or utilizing gamma rays or even particle beams to destroy cancer cells by damaging their DNA. It is typically utilized by using a machine that focuses that specific radiation precisely on the tumor or even internally with the use of radioactive substances such as actinium, and is either placed or within the tumor. And so it's just another form of treatment that just works to target the cancer cell, and it just targets the cancer cell by using these high energy beams and these high radioactive frequencies in order to break the cancer cell apart and mitigate it. While chemotherapy and radiation therapy are mainstays in cancer treatment, their lack of specificity can cause widespread damage to healthy tissues. While chemotherapy and radiation therapy work extremely hard to stop the cancer overall, because of their widespread ability and how fast they're able to spread throughout the body, they cause many problems, and many problems present themselves as a result of these treatments. One big problem is hair loss. And so when you're using these chemotherapy drugs or these radiotherapy drugs, they flow throughout the body, and while they do target the cancer cell, the possibility of the accumulation of this medicine in tissues that are normal, it increases. And so your hair cells and your pores can accumulate these drugs, and as a result of the accumulation of these drugs, you can lose hair and your hair can fall off. Another side effect is fatigue. The widespread and just the overall broadness of these drugs can take a toll on the body and just increases overall bodily fatigue. Another side effect experience, another more common side effect, is nausea, diarrhea, and vomiting. Because of how strong these drugs are, they're very damaging on the mucosal membrane layer of the stomach, and as a result, it upsets it constantly throughout your treatment. And another result is just overall just mental fog. These treatments are just so strong on the body that they have very hard impacts on the brain. And so, whether it's in the form of anxiety, mental fog, even symptoms such as depression can arise as a result of taking drugs such as these. And there are a wide range of effects. This is just the bigger scale of some of them. There's a lot of effects out there. And really, to improve the specificity, current targeted radionuclide therapies, which I'm going to refer to as TRTs throughout this talk, work to improve specificity in the cancer treatment. So what you see pictured is essentially a TRT. So we have a prostate cancer cell that has a receptor, in this case it's the PSMA receptor, and this receptor works as a specific binding site, a specific target that we're going to use to target to treat this cancer cell. And so it's pretty much we build a protein that's able to bind to this receptor. It'll pick up on the receptor and bind it. But the protein is going to be carrying a radiotherapy drug, some kind of radioactive element that's able to emit alpha or beta particles. And what it's going to do is when the protein binds to this receptor, the protein is going to utilize this radioactive element and target only that cancer cell specifically. And this is going to work to expand and improve on the cancer treatment in that way. And so we'll only target the cancer cell by binding specifically to that cancer cell. As I was researching and looking into all of these articles, one excerpt that I ran across is stated that one of the challenges of targeted radiopharmaceutical development has been achieving a balance between maximizing the absolute amount of radionuclide that can be delivered to the tumor and meeting the requirement that the tumor to normal organ dose ratios be as high as possible. And so pretty much there's a problem with the TRT. There's a big problem, and that the problem is that we have to find a way to deliver enough radiation while at the same time not accumulating too much of this drug in the tissues. Because the buildup of this drug in healthy tissues is very detrimental on the body. So finding a way to combat and find balance between that is one of the biggest hard-hitting questions in TRT therapy. And so going off that, I chose the article titled Covalent Proteins as Targeted Radionuclide Therapies Enhance Anti-Tumor Effects, which was worked on, researched on, and written by Li Wang and his cohort at the University of San Francisco. I chose this article as cancer is one of the biggest problems in this society, and finding ways to find a specific treatment can have worldwide implications. And so I decided to go with this article. One of the first things that Li Wang and his research group noticed is that covalent protein TRTs solve the safety efficacy dilemma by irreversibly binding tumors without altering circulation time. And so you see on the left-hand side, which is the base and current standard TRT, you have a protein that with a radionuclide. And what happens is you have a irreversible, or not an irreversible, you have a reversible binding of this radionuclide to the target protein on the cancer cell. While this is effective, it being reversible presents problems. And so since it's not constantly staying bound to the cancer tag site, that it can reversibly go the other way in the reaction and it can detach from the target site, leading to accumulation in the bloodstream and across healthy tissues, which we do not want if we're trying to have a more targeted cancer treatment. And so what they did is they found a way to make a covalent protein TRT. And so on the right-hand side, you see this covalent antibody, which is labeled with the FSY tag that allows it to interact covalently with a specific molecule on the HER2 protein. And so the FSY, which is pictured on the covalent TRT, is able to form a covalent bond between the HER2 protein, which is the target site on the cancer cell, and it forms an irreversible crosslink. This irreversible crosslink is huge because it ensures that the bind between the nanobody and the cancer cell stays intact, meaning that this radionuclide is only going to be targeted onto this cancer cell, which is very huge. So how was this done, you ask? The FSY tag enables site-specific covalent bond formation between the nanobody and the HER2 through suffix chemistry. And so how this works is the lysine is going to act as an electron-rich nucleophile, and it's going to attack the sulfur atom on the FSY. And so it'll attack the sulfur atom from the backside approach, and it's going to kick off the fluorine, which is a really good leaving group. And so what happens as a result of that, it forms a tetrahedral intermediate, which collapses, and then it forms the covalent interlocked crosslink. And this crosslink is very important because it just ensures that you'll have specific targeting of the cancer treatment and reduced toxin buildup throughout the body. So this is very important, and it's a very effective way of formulating this covalent bond, which would be very important in targeting these cancer cells specifically. So the FSY was successfully integrated into a HER2 targeting antibody with the use of genetic code expansion. And so Li Wang and his researchers, what they did was they utilized genetic code expansion to integrate this FSY protein into the nanobody. And so what they did to confirm that this FSY was not only added to the nanobody, but that it was able to form the crosslink with the HER2 is that they used mass spectrometry. And so in mass spectrometry, if the bond wasn't as strong, you would see a bunch of fragments. You see a bunch of fragments that span the whole picture, and those rays from the mass spec would break apart any not strong enough bond. But here you see a very strong and a very abundant peak at 13,767. And so that signifies and it shows that the crosslink was effectively formed and then that their strategy was very effective. Furthermore, the engineered nanobody cross-linked specifically and irreversibly with HER2. And so with the use of a cell culture, they utilized the HER2 protein on the cancer cell. So they grew a HER2 protein in a cancer cell, and they gave it the radionuclide that is non-covalent, which is listed in the top right. It's the gray with the radionuclide, the gray circle with the radionuclide. And then they also had the HER2 in the presence of the covalent radionuclide, which is the red nanobody with FSY tag and the radionuclide. And what they observed is only in the presence of the covalent TRT was a crosslink observed. And so when the non-covalent radionuclide TRT was implemented into the HER2 culture, that no crosslink was observed. And you can clearly see it by the arrow I have pointing to the western blot peak at 140, that the crosslink was broadly and very strongly observed at that peak. And it showed that only the covalent engineered nanobody cross-linked and formed a strong enough bond with the HER2 tag. They also realized that the covalent nanobody binds HER2 selectively in these cancer cells. And so on the first figure right here, what they did is they grew cells in a culture. And so they grew these cancer cells, and then they had two that were grown with the covalent, or no, they had two that were grown with the non-covalent TRT and three that were grown with the covalent TRT, and they allowed them to react and grow. And so what they observed was through this plotting is that only the covalent TRTs that had the FSY tag that allows for the covalent bonds, those were the only nanobodies that observed the formation of the crosslink. Regardless of the concentration that was used, as long as the FSY tag was present on that nanobody, it allowed for the crosslink to occur. Moreover, they also did a similar experiment using a cell culture, and so they grew cancer cells two that had the specific HER2 site, which are the two on the left-hand side, and they grew two that lacked that HER2 site, and then they grew it in the presence of the covalent nanobody. And so what you see is on this Western blot that only the ones that had the HER2 tag formed that specific crosslink between the HER2 tag and the FSY. And so only in the presence of the HER2 tag will the FSY form a covalent bond, and that is very huge because it shows the selective ability of the covalent antibody. And then lastly, they did this in live rats. And so what they did is they have the wild type, which is essentially the normal or the functioning form of the nanobody found in nature that lacks the FSY, and they have the engineered nanobody that has the FSY. And they targeted cancer cells that were in mice. And so they injected it, and they injected both the wild type into some of the mice, and they injected the FSY into the other portion of the mice. And only the mice that were injected with the covalent modified TRT observed the crosslink. And it just shows that this crosslink formation is specific and that it's only working to bind these HER2 cancer cells. Their research also revealed that the covalent antibody showed significantly increased radioactive retention in tumors for over 72 hours, with specificity being the biggest goal of their work. This work right here is very crucial. And so what it shows is PET scans of the cancer cells being targeted in both with the wild type antibody, which is the 2 rows up top, and then also the cancer or tumor cells being targeted with the covalent antibody, and that is listed at the bottom. And so you can see more specifically in the PET scan that's listed in the second row, the retention is shown by the color. And so in the wild type or the non-covalent TRT, the TRT loses its effect around 10 hours, which shows that it's not really staying in the tumor. It's getting to the tumor and it's leaving the tumor in a very short time. But with the covalently modified FSY, you can see with the PET scan data that it's retained for over 72 hours. With 72 hours in comparison to 10 hours, that's a really big difference. That means you're targeting not only this site specifically, but you're targeting it and you're retaining it in your cancer cells for a longer time, which shows that the covalent treatment is better at keeping this medicine only at the site of the cancer spot. The covalent antibody also significantly suppressed tumor growth. And so as you can see on this graph, what they did is they monitored tumor growth in the presence of both the covalent FSY tag nanobody or the non-covalent wild type nanobody, and they also monitored tumor growth in the presence of saline to serve as a control. And so what they noticed is that in the presence of saline that you have very increased and very fast tumor growth, which is expected because it's utilizing saline. But the non-covalent TRT, when it was utilized, had very similar effects. And if you're supposed to be a cancer drug, why is your cancer drug not performing significantly better than using saline, which is very problematic and is very not good in cancer treatment? But what they notice is that this covalently modified TRT with FSY tag is able to significantly decrease the rate of tumor growth and tumor volume that you see on the left-hand side, and that it keeps tumor volume very low in comparison to both the non-covalent TRT and the use of saline. And then on the right-hand side, it shows you tumor weight, and it shows you the same thing, with the orange colors corresponding to the FSY tag protein. It also not only keeps tumor weight or tumor volume significantly lower, but it also decreases tumor weight, which is showing that the covalent nanobody is significantly suppressing tumor growth. Its potent anti-tumor effect, covalent TRT showed no signs of systemic toxicity. So what I have here is scans of the liver tissue, the kidney tissue, the heart tissue, and bone tissue, and both where the nanobody that is covalent was treated, which is listed by the row on the bottom, and also the nanobody of the wild type, which is shown by the rows up top. And you see that there's no difference in the amount of systemic toxicity in between the two tissues, which shows that there's no negative side effect of utilizing this covalently modified TRT, which is huge in cancer treatment. And so overall, the success of covalent TRTs opens the door to a new class of safer and more versatile cancer therapies and beyond. Covalent TRTs represent a major leap in targeted cancer therapy, combining high tumor specificity with minimal toxicity. This platform also paves the way for broader, safer, and more precise treatments with potential applications beyond oncology. Thank you.